So good evening, ladies and gentlemen. Good evening. Hello. Welcome to the inaugural lecture of Prof. Robbie Pott. We are now about to start the event, so can you please stand as we welcome the procession inside the venue. Thank you.
Good afternoon, ladies and gentlemen. You are welcome to sit down. Usually I forget to say that, but today I remembered. I want to start by welcoming everybody that's here to this auspicious event, specifically the members from the Rectorate, Professor Sibusiso Moyo, the DVC Research, Innovation and Postgraduate Studies, as well as Professor Richard Stevens, who is the acting DVC Learning and Teaching. We also have Professor Bertie Fielding here, who is the Dean of the Faculty of Engineering. Professor Sorry, I was promoting you, but now I have to demote you the Faculty of Science. Sorry. Professor Kanshu Rajaratnam, who is the Director of the School for Data Science and Computational Thinking. Professor Celeste Viljoen, who is the Vice Dean Teaching and Quality Assurance in our Faculty of Engineering. Professor Corny Skutte, the Vice Dean Research and Industry Liaison. And Professor Christy Dorfling, who is the Chair of the Department of Chemical Engineering. But most importantly, our honorary speaker, Professor Robbie Pott. So thank you very much for being here, Robbie. Without you, it would have been a bit of an embarrassment. Also, Professor Pott's partner, Ian van der Westhuizen, as well as Professor Pott's parents, Judy and Ricky Pott, and his family who are joining us online. So welcome to you online, as well as the parents-in-law, Saki and Andri van der Westhuizen. Baie welkom hier so by ons fakulteit. Then also the funders and the collaborators in the audience, as well as those joining online, the colleagues in chemical engineering and the faculty of engineering, friends, collaborators, students, and all the others attending in person and online. And hopefully now I have thanked everybody that I was supposed to thank. I have some order arrangements that I have to share with you. So first, there's a requirement that you should switch off your cell phones. Also, if you need to go or attend to the bathrooms, it's out that door, down the left and on the left-hand side. In the event of an emergency, we have a very fancy system here. There will first be a voice that says there is an occurrence that they are investigating, so we don't have to run yet. But when they say evacuate and the siren goes off, then we do have to start running. You can enter the room by that door, and there's a fire escape just to the right when you go out, or you can exit there, and there's stairs there, and then you can exit via that. So hopefully with that we are safe. So it's my honor and privilege to introduce Professor Robbie Pott. He works at the intersection of chemical engineering, molecular biology, bioprocess engineering, and biotechnology. He's a professor of chemical engineering in the Department of Chemical Engineering at Stellenbosch University. He completed his undergraduate degree at the University of Cape Town, and then he read for his PhD at the University of Cambridge. So here we do PhDs, and we just read for them, so it's fantastic. But he was under the supervision of some very famous professors, Professor John Dennis in chemical engineering and Professor Chris Howie in molecular biology. His doctoral research focused on the conversion of waste glycerol into hydrogen using photosynthetic bacteria. Upon completing his PhD, he returned to South Africa to join the University of Cape Town as a postdoctoral research fellow. In 2015, he moved across the peninsula, or across the Liesbeek, as we usually say, to Stellenbosch University to take up a lectureship, and he has grown a significant research group in chemical engineering in the years since. As postgraduate supervisor, he has guided 26 master students and 6 PhD students through to graduation, and he currently supervises 8 master students and 5 PhD candidates, and is the mentor to 2 postdoctoral fellows. So for those of you, those are impressive numbers in the Faculty of Engineering. So well done, Robbie. He has served in several administrative and service roles at both departmental and faculty level, including as postgraduate coordinator for the department, academic head of a focus area for the Structured Masters Program, and head of the Faculty of Engineering's Innovation Council. His research straddles the boundary between pure academic work and science capable of being commercialized. While he primarily focuses on bioprocessing engineering, he also has a strong interest in engineering education. And I can attest to that. We have a cohort of very, very active members in the faculty who do lots of innovative things in teaching, and learning and teaching. So you should come and visit some of our events here, Richard. And Robbie is definitely a shining light in that. So much so that he received the Engineering Teaching Excellence Award in 2019. And in 2025, this year, he received a ROCTA, or ROCTAS. It's a recognition of contribution towards academic success. So Robbie, you are not only an accomplished researcher and academic, but you're also a great teacher. And we really cherish having you in the department. And we are looking forward to listen to you for your inaugural address. The floor is yours.
Good evening, ladies and gentlemen. Thank you very much for the warm welcome, Vickers, and thank you very much for the attendance, the academic procession, and everybody in the audience and online. This evening, I'm going to be presenting some of the work that I've been doing in the last 10 or 15 years, but I'm cognizant of the fact that many of the people in the audience are not engineers or scientists necessarily, and so I'm trying to present it in such a way that everyone can be interested and hopefully intrigued. So if you have difficult questions about the context and the application, you can ask me afterwards. So we start by thinking of me as a little chokurki. When I was in school and deciding what to do, there was a diverging path ahead of me. I was deciding between medicine, music, or chemical engineering, the third of which seems like it doesn't necessarily fit with those other two, or maybe none of them fit together. But an opportunity arose where I did the Science Olympiad and I was invited to go up to the University of Pretoria, even further up the peninsula. And we visited the nuclear facility out there, Pilindaba. And I got to see the Cherenkov radiation there in the reactor, glowing blue in the water. And we met a whole bunch of researchers there, and one of them was a chemical engineer making radioisotopes for medicine. And he convinced me, he didn't directly, but thinking about the work that he did, he convinced me that one of the best ways to improve the world is by creating new things that other people can apply and to create new stuff. And so I decided, let's do chemical engineering. And so I came down to the University of Cape Town as a slightly bigger chokurki. And some of my colleagues from ECT are here or attending online. And my hair was even bigger than that picture indicates. Then I went across the water to the University of Cambridge to do my PhD there, which was incredible and an amazing opportunity. And so I studied here at this beautiful old building. I did some crazy things like competing for England in formation dancing and splashing on the water a bit, but mostly I did this. And I don't show you this just as a show and tell pictures of my youthful exuberance, but rather as an indication that research and developing ideas is not just about hard work, although it is that, and being in the lab, although it is that, but you need to get inspiration from somewhere. You need to develop your ideas and communicate them with people who come from various walks of life. And in all the types of projects that I'm going to show you now, they never arose simply from me sitting and thinking. They always arose out of conversations and being in different places. And so it's very strong in my mind that if you want to be a good research engineer, you've got to go out and look and explore and be interested. So I did that, but mostly I did that. And then I came back to ECT for a little bit, and for the most part I worked on making blue food coloring, which sounds silly, but was lots of fun. And then I got called across the peninsula, as you say, to come to Stellenbosch University, and maybe I'm looking a little older, and any second I'm going to look like that. So let's think about what is the stuff that I do that I'm spending my time on. So I call myself a bioprocess engineer, although my degrees are in chemical engineering. So bioprocess engineering kind of takes the idea of the biological material, the biological organism, and applies the principles of chemical engineering to it. Can we take biological process, biological material, and make valuable stuff out of it? So for the most part, people think that it looks like this, a bioreactor. Maybe you put an organism inside there, maybe it ferments some beer. But in reality, it is more than that. We don't just do that. What we want to do is a complex interplay of, is there value? So we need some kind of creature to make something. Maybe it's producing an individual component. Maybe we're taking biomass and extracting something. We've got to feed it on something. We've got to grow it, and then we've got to separate it out. And hopefully there's some money at the end as well. Sure. But this is not a once-through process. It is continuously and always iterative. You never start with something that is going to be your end product. You always develop, change, modify, try again. And so there's a real aspect of playing with the process until it does something interesting. And it doesn't always do something interesting. Often there are failures and it doesn't work. And that is also useful and a part of what we do. For the most part, a lot of people think that we simply sit between the intersection of can it work and is it going to make us some money? Which is fine, and that is what we do. But there's a third dimension as well, is how does this interact with society as a whole? How does it interact with the environment as a whole? We can't do those things in isolation. We need to do them all together. And which is why I say it is an iterative process, because solving all of these three things at the same time is not a trivial task. So I'm going to talk about some of the research that I've done over the last few years. And I've had many graduate students over a decade and more than a decade, and each of one of their projects is interesting and worthy of an entire hour's presentation. I'm not going to subject you to that, although you have sat down and you can't leave until I'm done. So I'm going to show some of the things that I do. And if I miss out somebody's research, I apologize. But if you're interested, we've got lots of publications and theses that you can look at. So my research is based on who is doing the work. It's this creature of some type, cyanobacteria, fungus, bacterium, yeast, whatever. Somebody does the production. What do we feed it on? Can we use a pure substrate like sugar, which sounds great, but maybe is expensive, or maybe we can use a waste material and produce something valuable out of it. What kind of reactor do we allow this thing to grow in? Does it produce well under normal bioreactor conditions, or do we have to design something entirely new? How do we get the valuable product out at the end, away from the waste materials? Can we recycle the waste materials to do something else with them? Is it viable in terms of the technology? Does it work? Often you'll develop a whole process, and then only at the end of many years of study can you realize that was a great process, it is not going to work. And that's also fine. Is it sustainable in terms of, are we making something that we should be making? Are we using something that we should be using? Is it economically, societally, and environmentally sustainable? And does it help our society move forward? Those are the kind of questions that we ask. And of course, it's not me just in the lab doing it. I need pairs of hands that are interested, that are driven and enthusiastic, who can spend the time and the frustration that it is to do laboratory work. And then at the end, or kind of halfway through, is we sit here in this bubble. Can we move beyond here and actually take it out into commercialization? Can we patent it or trade secret it? Can we partner with industrial partners so that they can actually apply the work that we do here? It's all well and good to develop new technologies in this building, but it needs to go beyond this building if it's going to have actual impact in the world. So in terms of this, I'm going to show you some case studies of work that we have done and think about which of these aspects are coming through in each of those projects. I'm not going to be able to separate them out, so there's some homework for you to do while you're listening, active listening. So my work kind of sits in this intersection of upstream processing, downstream processing, can you separate out the material, bioreactor and bioprocess design, how do you make it, how do you integrate it, and sometimes we use tools like modeling to do that, and then not an orphan child but fully integrated into everything else that I do is there's these pairs of hands that we use to do this work that get developed in this process of doing the work. How do we make sure that they are becoming excellent engineers and research scientists in their own right? So in terms of work, I'm going to tell you a little bit about photofermentation, which includes some reactor design, some computational fluid dynamics, some molecular biology, don't get too afraid, engineers, it's okay, some material science. We'll also talk about some different reactor design for a different set of organisms that are doing biosurfactant production. We'll apply those biosurfactants, we'll do some separations. I'll talk about some specific materials that we can convert into valuable things. And there are lots of other projects that kind of sit there. And integrated throughout it all is understanding how our students learn, how our students develop themselves, how they grow, how do we become good engineers. So let's start with the first case study, biological hydrogen production. This was kind of the work that I had started with during my PhD, and several of my research students have continued it. The background is maybe we don't want the whole world to be in red. Let's alternatively find different sources of energy. And the idea for this is there are many waste materials that come from industry, that come from normal human occupation. How do we capture the energy that's inside that? Can we develop a bioprocess that converts those materials into something that is useful, in this case hydrogen? And hopefully we get some money out of it, but also hopefully we get a treated waste out of it as well. Now, if you ask, any engineer in this building, what's the best way to make hydrogen? They're probably going to answer, well, let's put up some solar panels and some wind turbines and we can use an electrolyzer, and that is almost always the right answer. But if we've got a waste material that is sitting unused and needs to be treated anyway, perhaps we can generate value from it at the same time. Maybe then it's worthwhile. So the organism that we use to do this is this pretty purple creature, which I completely chose because it's a nice, pretty color. And we apply it to wastewater conversion. I started on crude glycerol, but we've looked at all sorts of things. We've done some molecular biology on the organism to try and improve its hydrogen production rates, because surprisingly enough, the organism doesn't particularly like being used as a little machine. It wants to grow and be happy. It doesn't necessarily want to produce lots of energy. Then what kind of system can we use to grow it in? And I'll show you some bioreactors that we developed and modeled. And we also developed some new materials into which we can hold the organisms so the wastewater flows past and they convert it, but they remain inside. Okay. So we'll jump in with a little bit of molecular biology. Don't worry too much about the picture. It's just illustrative. The organism doesn't necessarily want to produce hydrogen in large amounts because it's energetically expensive for it. So what we did is we figured out which enzymes it is using to produce hydrogen, and we increase the amount of those enzymes that are produced via some Pott promoters. And what we found is, great, we produced lots of enzyme, a significantly improved amount of enzyme that was inside the cell. In terms of the hydrogen production rate, didn't change at all. A failure, but an interesting failure. So we know that it's not the enzyme that's the problem. It's probably something else. It's the work we've done since, it's narrowed it down that it's almost certainly the electron transport chain. The organism is absorbing lots of light, but then is not managing to funnel it towards these enzymes. So the work continues. Then we investigated how do we keep the organism from washing out of the reactor. So we've got a wastewater that we're flowing through a container. The cells want to go with it, but we don't want them to. They grow comparatively slowly, but we want them to stay as a catalyst inside the reactor. And so we developed what we call PVA cryogels or hydrogels, polyvinyl alcohol, very similar to the white glue, the wood glue that you use to stick material together. And the organisms are very happy inside of this material. And it also means that we can keep the organism inside the reactor. So this is what that looks like at a microscopic level. The little red things are the organism. The green stuff is the immobilization matrix. In the first case, you can see there's lots of porosity, these black holes surrounded by green. What that does is it scatters the light, which we don't want. So we developed a new material which now looks completely black to your eye because it's a monolith. It doesn't interfere with diffusion, but it does prevent light from scattering, and it does not allow the organism to escape. So you managed to solve a tricky problem in terms of you need a material that is biologically compatible. It allows for diffusion of a reagent in and product out, keeps the organism there. Is the organism happy? We've left the organism inside these materials for, I think it's a year now, and they are as happy now as they were when we put them in, which is surprising. In terms of this material, we developed, we needed to have transparency, so it allows light to penetrate. We compared different materials in order to protect the organism from the pretty harsh conditions that the immobilization entails. Right? And so we did something like that. And for the most part, it's based on polyols, which allow the polyvinyl alcohol to make that transparent monolithic structure. So now we've got our organisms entrapped in a solid material. They're happily chugging away and producing hydrogen. In what reactor do we do it? Now, because we are producing a comparatively low-value material from a low-value material, we don't want to be inputting energy in terms of mixing. We don't want to be having an expensive little glass reactor. We want it to be sitting outside in the sun and let it do its own thing. So we developed what we call a thermosiphon photobioreactor, which is a naturally circulating reactor based on incoming light. So the light comes in, it heats up the fluid, it allows the fluid to circulate. So the organisms are circulating inside the reactor by itself. And we did some clever CFD. The poor student who did this, when she started her PhD, said, please no modeling, and then COVID happened. Sorry. So we did some clever things. And you can see I mean, pretty colors for CFD, it always looks good. But the interesting thing is that we do manage to get good circulation from this incident light. The next question is, okay, well, light is coming into the reactor. Is it enough? What is happening to the light? And so we modeled what happens to the light as it enters the reactor. Is it absorbed? Is it reflected? Is it scattered? And the important thing here, don't worry about the maths, is that, except for the mathematicians in the room, you're welcome to worry about that, is the scattering term. Is that light still available for utilization by the organism after it's been come into the reactor but no longer going in the same direction? And the answer is, well, yes, it is. But the organisms are dark. They absorb a lot of light. So if we have our normal reactor, so here's the light source shining this way. Here's lots of light being absorbed by the organism and darkness for the second half of the reactor. Darkness means no hydrogen production. So we modified the reactor and said, well, can we do some easy reflective things? That seems like a sensible idea, and yes, that works. And suddenly we have light penetrating even to the center of the reactor. And we're getting also better than heat transfer so that the reactor circulates more easily. Okay. So that works nicely. But again, we now have this core that's not so bright. Can we maybe modify these reactors so that we get axial mixing rather than just vertical motion? So we included some static mixers, and they work fine, except the organisms love to stick to them. So we went from a nice thick culture circulating beautifully to these things, a beautiful pink color and the fluid clear. So sometimes it works and sometimes it doesn't. But all of this is not meaningless, but underpinned by, are we going to make any money after doing this? And the answer is, well, no, not yet. We need the organism to operate much faster. We need it to be able to be reused for much longer. And we understand that only after you've done the sets of experiments to set up the data. So that's the frustrating thing, is you can't tell at the beginning if your idea is a good one, but it is also the exciting thing. As you go through the process and you understand it, and at the end you figure out, okay, can we sell it or not? And sometimes we can and sometimes we can't. The next topic that I want to talk about is, instead of using an organism, can we grow something and then extract something valuable out of it? So this is an AI image of, let's take a waste and make money out of it. Thank you, AI. I wouldn't have invented it like that, but yes. So the kind of examples I'll do here is, we started with wine wastes. You press the grapes, you make some wine. There's a material in that wine waste called resveratrol, which apparently is going to make us live forever, going to keep us young looking. People will pay for it. It stays in that waste. Can we extract it? So I developed a process to separate, extract, purify that material out, which we've got a patent on. And hopefully somebody will buy it, anybody listening. We'll look at some malignous organisms, making oils and waxes out of raw materials. Specifically, I'll look at seaweed processing, because that's quite fun, and cannabinoids, which I'm sure you're all waiting for. And then maybe we'll look at some metals, or maybe not, I don't know. So, taking a biological material and converting it into something else is not so trivial. A seaweed, like this stuff that you see on the beach, kelp, is comprised of hundreds, thousands, millions of components. They're all there mixed together, the organism has produced them. Not all of them are necessarily valuable, not all of them we want. In this case, we were looking for what we call alginate oligosaccharides. So alginate is a structural polymer that makes up the structure of the seaweed. It's cellulose but flexible, which allows it to not be broken by the sea. Alginate oligosaccharides are bioactive. If you spray them onto your plants, they will grow nicely, they produce lots of flowers, they make fruit. Sounds like the farmers will pay for that. So our idea is, can we collect this material and either using an enzymatic method or a chemical method, hydrolyze that material down into alginate oligosaccharides. I'm not going to tell you about the chemical method because the company paid for it, and so it is a secret. But I can tell you about the lyase method, the enzymatic method, because nobody has paid for that yet. So the idea is we've got this long structural polymer. We want short pieces of it. That's where the activity lies. We went out, well, not we, one of my students went out, and caught some rotting seaweed and some sand fleas and some all sorts of creatures. And those organisms are able to eat this, so they must be able to break it down. And we isolated 20, 100 different organisms that grew on it. We figured out which ones of those are able to hydrolyze alginate down. We've since isolated and produced the enzyme and are now taking those enzymes and trying to produce them in a different host, E. coli or Pichia or some other organism, so that we can produce lots of the enzyme. Because again, the organism that eats this stuff doesn't particularly want to use all its cellular machinery to make the product that I want. But I can make other things do that, whether they like it or not. In terms of an application of the similar kind of material, now we move to the bioscience side. If we combine alginate hydrogels, carrageenan hydrogels, and what we call an oleogel, which looks like a thick paste, it's basically oil that's mixed with like beeswax or something, it's pretty solid. If you mix those things together, we can develop a slow-release drug mechanism. So the idea is, this is a person, inside of a person. You eat this drug, it goes into the stomach. If it was just a pill, it would have been released there. In this case, the medicine that we want to deliver is praziquantel, which is used to treat bilharzia and other worms. The worms sit here on the outside of the colon. Thank you. We don't want it to be delivered in the stomach. So we use the hydrogels, the alginate and the carrageenan, to protect the praziquantel and the oleogel. Then it passes into the intestine, and the pH changes, and different enzymes are released. And the alginate and carrageenan is dissolved, broken away, and we're left with little particles of oleogel, which then cling to the walls of your intestine and slowly release the praziquantel. So this is an application of the same kind of material that we could break down and use in agriculture. We can also use it in medical bioscience. But we can only do that once we understand what the material does and how we can play with it. You can only play with it if you have tried. Another example is the nascent industry of cannabis bioprocessing. So there's several production facilities in South Africa now that it's been semi-delegalized. Where's a lawyer? They produce this material as a waste, a winterization wax. Inside the wax, they did not know what was in there. They just kept it in containers, hopeful that one day somebody would figure out how to do something with it, or disposed of it at great expense. Incinerated, biological waste, medical waste. Inside that material, we found alkanes, which are interesting, fatty acids and fatty alcohols, which are interesting, sterols, which you can do fun things, but a significant portion of it was the product that they were making, the cannabinoids. They were losing about a quarter to a third of the material that they were processing into this wax. They didn't even know. So we developed a system, again, we being the me collaborators, clever students, hard work, not necessarily my hard work. We developed and patented two processes for separating out the cannabinoids from this material, one based on solvent extraction and anti-solvent precipitation. In this example, what we do is we dump our wax into a particular set of solvents. It dissolves everything. Cannabinoids, waxes, everything goes into solution. Then we add enough water that the waxes no longer want to be in solution, and they come out, they precipitate. But the cannabinoids have a slightly different set of properties, and so they stay in solution. So then we can catch them. And we can purify them based on that. We've got another superstar in the audience who found some mushrooms in the field. If he offers you a mushroom, maybe say yes. So we've got a pleurotus, a kind of oyster mushroom, and a type of hericium that he found in Neisner. These organisms are interesting because they're growing in South Africa, but they're also interesting because many of these have interesting components. But we don't know what the interesting components are until we fractionate and test them. So we took these materials, did a simple extraction, a simple set of separations, and then a not so simple set of tests to see whether there is any activity or not. And amazingly, I keep telling Brayton this, it's a little bit of a miracle, there was activity against cancer cells but not against healthy cells. So we were just fishing and we found it. The process is interesting, but also now the application is interesting. So that's useful. We find an organism not been classified before, not been investigated before. We separate out some particular components, look for the activity, and once you drill down, you find activity. Now we need to go to the medical people and say, okay, well, how much do we add? Do you want to take some? Maybe they'll say no. So while we're talking about separations, it's a core component of what chemical engineers do is to take a raw material which is comprised of many things and do some magic on it so that we are left with valuable material on one side and other stuff on the other side. So I have another piece of kit that I'd like to tell you about, the simulated moving aid chromatography column, which looks like a black hole machine, and we also call a water feature because it leaks like hell. But the basis of it is this. It's chromatography. So we add a mixture of materials at the top here. Down it flows. Some goes fast and some goes slow, which means that we can separate our different materials. So if we have such a column, that's useful and we apply it. But as a chemical engineer, I don't want to do one. I want to do it continuously. So can we modify this single column into a continuous operation where we're always getting out on one side product A and on the other side product B? And the way we do that is we move the bed. So we don't just run this through, we allow the bed to go up. And the principle is this. So let's have two racing cars. Well, a racing car, which goes fast, the thing that went down the column quickly, and a normal car that goes slowly. And then we allow the belt here to move. If we have the belt too slow, both go into the same bucket. We've not done a separation. If we have the belt too fast, it pushes both cars, even the speeding car, to the other bucket. So we choose one where the belt is faster than the slow car, but slower than the fast car. That's what that looks like. And then we can continuously separate these materials. Unfortunately, this belt is not so easy to do in reality, because then you have to have a continuously moving solid substrate. So what we do is we divide a simulated moving bed chromatography column, where we have columns and we switch them. So what happens here, I know it looks like craziness, and it is a little bit. We have fluid flowing in one direction, this way, anti-clockwise, and columns moving in the other direction, clockwise. So the fluid flows one way, the belt flows the other way. And if you do it well, then you get separation. If you do it badly, then nothing happens. And we can use this for separation of all sorts of material, as long as there's interaction with a resin of some type. We could use it on metals. What we are using it for is sugars. We could use it for the oligosaccharides that we made a little bit earlier, if you want to fractionate it specifically. The second to last kind of case study I want to talk about is biosurfactants. Surfactants are soapy materials. Organisms produce soapy materials for a variety of reasons. We have one class called lipopeptides, lipo from the fatty part, peptides from a protein component. These are produced by lots of things, but we get it from bacillus. They live side by side with fungi and so forth on fruit, and they want to fight for space with the fungi. So they produce these things, which then kill the fungi. You can see an application already. We can use it then to spray these lipopeptides, which are edible and safe, onto fruit after we've harvested it and while we're shipping it to Europe, and that will stop them from rotting. So that's one class. We also have mannose erythritol lipids, try and say that 5 times fast, and cellulose lipids, and those are produced by a different class of organism, fungi and yeasts. And they're useful because they can be applied to the skin, and it, again, makes us look young forever. There's not a theme to my work or anything. And we can do clever things like novel separations and novel bioreactors so that the organism produces lots of them, or we manage to separate them out in a particular way. So this is what those lipopeptides look like. They've got this nice big ring structure and then the fatty acid component. And these things will pierce into the sides of the fungi, stopping them from growing. So we can stop your fruit from rotting. And actually, you can buy this stuff already in the supermarket as a normal detergent. If you look at those biodetergents, they're using biosurfactants. But it's not so easy to catch them, because the organism that produces them, as soon as it runs out of food, eats them again. And we can't detect them quick enough to know when it's about to run out of food. So we had to devise a new way of separating out these materials, and the way that I landed on is what we call aqueous two-phase separation. So we have these two layers, top and bottom phase. It's both water-based, which means that the organism can grow and live inside it. So the organism lives down here, and if you choose the conditions right, then the product goes into the top phase. So you have separation from the product and the organism producing. So then you can have something like an in situ fermentation, where it's continuously producing, you can continuously separate the product, make some money from it, get the organism to continue. But there's clever stuff that you can do with this aqueous two-phase separation. You can separate biomolecules, such as we are looking at RNA and DNA for vaccine production. It's a tricky part. It's very difficult to separate RNA without destroying it. So this is now a gentle system that can separate it. Or we can separate the biomolecules from, for instance, the organism that is growing. And if you apply it well, you can then design a bioreactor around it. So this is a semi-partition bioreactor. Semi-partition because there's a space here that allows the settling of those two phases. The phases are mixed, the organism is contacted with both phases in the normal part of the reactor, and then we take the top phase off, which now has the product, separate it out, and then we're left with product continuously accumulating in this outside portion of the reactor. And we did obviously some modeling on that, so there's another colorful pictures. These compounds also have interesting activity with metals. So it has two carboxylic acid groups that can make a little bridge, which then can interact with metals, causing them to precipitate. So if we have mine wastewaters or mine process waters that have metals in them, we can then precipitate out, in this case, copper, nickel, and cobalt, but we've also done it with other things, some of them heavier, some of them lighter. And again, the science is driven by making pretty colors. Right. So they're useful not just in the fruit application, but in other things. And we wouldn't know this unless I had a conversation in the tea room saying, I wonder if there's activity. Let's go test it. And one of my colleagues, who is now an expert in the metals part, and me, who knows a little bit about the biosurfactants part, then we work together to make it work. In terms of the manosyl lipids, they're interesting and useful. They've got these different groups attached to them. And what's useful is they have a few groups that are available for further reaction. So we can take an organism-produced product, do some clever chemistry. I'm not good at the clever chemistry. I speak to the chemists to get that done. Thank you, science faculty, for attending. But we can do that and then get different properties. So we can combine both a biological approach and a chemical approach to make new materials. And they are useful. They look very similar to a structural component in your skin. So if you apply it to your skin, it helps it heal faster. So that's useful. But the organisms that make it don't necessarily want to make lots of them. So we've been trying to design different types of reactors, different types of separation systems that then catch or increase the productivity of these components. And this is what that looks like. Kind of gross. Or delicious, depending. The organism that we use for this, or Stalagomatus, actually is an edible fungus that people, particularly in South America, it infects corn, it makes a gross-looking thingy. Delicious. I wouldn't necessarily eat this bit. The last thing that I'd like to talk about is what underpins everything. I'm not simply a researcher. I must do both research and teaching, even as just doing research. I take on postgraduate students, freshly minted engineers and scientists, and we've got to develop together the ideas for their projects. And so I really do see myself, and I think it is an imperative that an academic sees himself as both a researcher and an educator. And we're not just an educator in terms of imparting knowledge, but also learning from our students as well. So again, here's what AI thinks that looks like. And I've approached this at multiple levels. So we have gone all the way down to some proper sociology of education. I'm not going to subject you to that now. But it's fascinating and it's useful. There are tools that are available for us to look at. But as an engineer, we are not necessarily taught any of those, but we can access them. One can continue to learn and be the student as well as the teacher. So the kind of things that I've looked at are using this scary-looking plane to understand how students learn, how students come to understand something, how they develop the perspective as an engineer, how they sort of develop their worldview, and how they approach things. I've also looked at post students, our students who have gone on to work, graduates, and tried to understand from them, are we doing things well? Are we doing things poorly? There will always be both of those. We are tasked with improving the bits that we're not doing great. I also am quite passionate about not simply making students do things because they're going to get marks. They hate that. They want to do it just for the marks because it's been transactional for them their entire school career, so why would it not be now? But in reality, in the world of work, it isn't. They don't get awarded marks for designing a bridge that's going to withstand 100 years. So we need to be training them for that, not for, please can I have one more mark for my exam? And I think a lot of what underpins that is grit, and a lot of what underpins a good researcher is grit. The other side of grit, of course, is knowing when to quit, which is a useful thing as well. As I say, at some point you check, is the idea a good one, or is it time to be done? And a lot of that is then to do with student success. So in terms of the academic, we now sit with our robes and our titles. We are taught how to be an expert in the field, how to have an impactful research program, how to take our work and to have it used by industry, how to get funding. These are all things that I'm evaluated on, as I should be. What we're not taught necessarily is how to be a good lecturer, although I think this faculty is doing a good job of improving this. Understanding how to run a course, how to impart knowledge well. What does success look like for a student? So I think that we need to remember this other side of ourselves while doing the work that we do as a research scientist. So I'm afraid what often happens is this. When you start, starry-eyed, the students are real people whose names you may know. They then become more and more amorphous. 10 years of teaching first-year chemistry to a class of 450, 350, 200, it's added up to several thousand students I've taught over the last 10 years. It's tempting to turn them into this, but that's not necessarily what we should be doing. I kind of view it like this. It is perfectly acceptable for you to sit here as an educator technician, to be one who does the work, teaches, maybe well, but there are depths that can be explored here. You are sitting on the surface, but there's lots underneath. You can try things, explore, reflect. Did it work? Did it not work? You can try to understand why did something work or not work. Peer into the head of a student or your colleague. And maybe eventually one of us reaches what we call a scholarly level, a scholarly teacher. And this is aspirational. I don't think any of us are necessarily there anytime. But we all try and make our contribution where we can. And I think that it is interesting to plumb these depths, as well as the depths of oligosaccharides and biosurfactants and others. There's lots here. So in ending, I'd just like to show some pictures of some of my students. It's not all. Some sent me some pictures of themselves and where they are, and some didn't, and many of my students are sitting here in the audience. But they're doing great. I particularly like Elze with her massive pile of foam, which she looks very proud of, but it's definitely not what's supposed to happen. Jackie's here in the audience. We've got students all over the world doing all sorts of things. Engineers, scientists, researchers, academics, working in industry or doing other things. And none of my research program could have been done without these people. And the people who I have not put up here are my colleagues and collaborators. I couldn't do any of this without some money to grease the wheels, yes please, and collaborators to challenge me on ideas and to discuss things and to bring different perspectives. So thank you, everybody. I won't name everyone because I will leave people out. So on that, thank you very much for your attention, and I open the floor to any questions. Everyone understood everything clearly. Well, if there's no questions, then again, I thank you for your attendance and for your interest, and I look forward to conversations afterwards.
Thank you so much for the lecture. So I think there was still a question. Some people wanted to ask, and maybe we're feeling shy. But maybe if there are no questions, I'll ask the first one. I wanted to find out, I see the intersection. Thank you for a great lecture. I think it was quite amazing. But I just wanted to check, so to what extent do you have to work with, say, people in the medical sciences? When you do your research, do you have students from the medical field who are in your research group, or fellow researchers? How do you go about that?
Absolutely. So there's no way, I mean, you can see the work spans all sorts of things, from agriculture to medicine and so forth. And I'm not a medical researcher. I'm not an expert in that. I need to work with people who are expert in that space. So we do take in students, and I have taken on students who come from medical background, microbiology background, chemistry, physiology, physics. All of those people are able to contribute by bringing their own perspective. And then we bring the engineering perspective as well.
Cool. Thank you so much. Any other questions? Are there any students here? Yes, please say your name and then the question. Do we have a mic, please? Thank you.
Thank you, Robbie. I'm Dr. George Decker, and I think Robbie's head of both PhD students. So I just want to ask nothing about all the presentation, but when do you think you're filled as a supervisor, and what are some of those challenges to young ones that are coming into the field to also be able to grasp the future?
Thank you, George. So your question is, where did I fail as a supervisor? You can see George has been trained by me, because you can learn a lot from where things didn't work. And it's easy to have a roadshow of success. But a more helpful thing is to have a roadshow of, can I reflect on what didn't work? So where I have failed as a supervisor is often sufficient time. Our schedules are full. And I wish that I had more time to dedicate to each of my students to talk through the work that they're doing and difficulties that they're having and challenges and what's working and what's not working. So that is one thing that I think we could do better. And as a young academic, that's, you've got more time, so use that time and to learn from your students as they go.
Okay, thank you very much. One last question. Anyone who's feeling courageous? Yes? Just tell us your name. Use the mic. Thank you. The question.
My name is Grayson. I'm wondering, what's been your favorite and least favorite
So Brayton's question is favorite organism, least favorite organism. So for those of you who haven't had to grow organisms before, they are notoriously tricky. They will do what they want. In order to get repeatable experiments, you have to read it poetry and sing to it, dab blood on the door, anything. My favorite organism is my purple bacteria, because it's been my baby for a decade and a half. But it's also very annoying and easily contaminated and grows slowly. But the mushrooms are fun because they're faster and you can see them. Yeah, they're good.
Good. Okay, thank you very much. I think let's give him a clap again. And just a few remarks from my side. So I was introduced. My name is Sibu Moyo, for those who may not know me. I'm the current DVC for Research, Innovation and Postgraduate Studies at the university. And I know that we were acknowledged by the dean as we came in. So as you know, first of all, before I go into I have got 10 pages of a speech. So no, it's much shorter than that. But what I wanted to just say is that I think today's lecture was quite amazing. I think you made chemical engineering sound so fascinating and interesting for everybody. And I'm sure if our first years saw what you're doing, they would be really attracted to the subject. So I just want to congratulate you on that. My background is in maths, so I was very happy to see at least one CFD equation. And I thought you'd spend more time on it, but we can talk about that later. So as you know, and I think maybe the other thing which I took out was the application to cancer, treating cancer. So we always struggle to understand, how applied, what can we do together around interdisciplinary or transdisciplinary research, but the research that shows even engineers can contribute to health solutions, right? Through different diagnostic tools, but also, I think for me, the cancer part is really interesting, and I want to ask another question because I wanted to ask who else we are working with in that area, but there's a lot of, innovation and also new solutions that I can see. So, as you know, for those of you who are here today and those who've joined us online, I just want to thank you for joining us, but also, as you know, the inaugural lecture, of course, for us as a university is really a formal event where we recognize our staff who've been appointed as full professors, and for them to showcase, to engage with their students, with the public in simple language, and you can see that the way it has been explained, even a layman can understand. How many of you here are from the social sciences, or how many of you are engineers in the crowd today? Engineers in the crowd? I know it's the majority, but the rest are not engineers. So I was saying, if you can explain the lecture so that your grandmother, your parents understand what you do, then it's really a big achievement, I think, just to be able to communicate the science. So, of course, we are really honored that the lecture that was delivered today, of course, from laboratory to lecture hall, developing bioprocesses and students, has provided, of course, for us an engaging and provoking journey through the many skills of work that you have showed us, of course, from the biological, technical, and also the applications to human health as well. So I've spoken about what stood out, but beyond your research also, I think your passion for students, the number of students you've supervised at your age. Sorry, we couldn't ask how old you were, but I tried to ask AI, but I won't tell you what it said while you were talking. But it's amazing what you've achieved, I think, at this stage. And I think the next is how you show the integration of your scholarship of learning and teaching and research is really magnificent, and an encouragement to those who also aspiring to become academics. So on behalf of the university, of course, I would like to really congratulate you on this important milestone in your academic journey. And also, we are immensely proud to count you among our professors, and we look forward to your continued contributions at the university, not only in advancing bioprocess engineering, but also in contributing to skills capacity building amongst the postgraduate students, working with other scholars, but also in shaping a more sustainable, knowledge-driven future for our country and continent. And I think we look forward to the breakthrough through the cancer research. I believe we'll have more patents and innovations. So congratulations and thank you so much.
Thank you.
Good evening, colleagues, ladies and gentlemen. I have the privilege of extending a couple of words of thanks to the people who have made the event tonight possible. First and foremost, thank you very much, Prof. Robbie Pott, for your presentation on the innovation and impactful research that you have conducted over the past decade and more. I think the presentation tonight again reminded me why Robbie is such a highly respected colleague and a very successful academic. Thank you very much, Robbie. I would also like to extend a word of thanks to Prof. Sibu Moyo, our Deputy Vice-Chancellor, Research, Innovation and Postgraduate Studies, for responding to Prof. Pott's lecture and for officiating the proceedings tonight. Thank you very much, Prof. Moyo. And then an event like this is definitely not possible with a lot of hard work and administration happening in the background. So I would firstly like to thank Corporate Communications and Marketing Division for their arrangements and also for recording and live streaming of the lecture tonight. And then specifically also to our Dean's Division and our Dean's Office staff, specifically Ulrich, Clint and Murray, who have done a lot of work to make sure that the venues are set up and other related arrangements have been made appropriately. Thank you very much to Robbie's family and also all the other guests attending tonight. We are really grateful for the role that you've played in Robbie's journey and also for being present to celebrate this significant milestone in Robbie's career. I would like to invite everybody to the cocktail reception afterwards. And to conclude our formal proceedings here, I would like to ask that you all rise and then please wait while the academic procession leaves the venue. Thank you very much.